X-ray absorption spectroscopy, we are studying extended X-ray absorption, fine structure, EXAFS. The EXAFS function is obtained from the X-ray absorption spectrum by subtracting the absorption due to free atom. This subtraction is done automatically. The absorption which is due to free atom, this is subtracted from other X-ray absorption spectrum, and in this way the EXAFS function is obtained. On the EXAFS data, a mathematical operation is performed. That mathematical operation is called Fourier transform. So when we apply Fourier transform to EXAFS data, it gives a radial distribution function. And what is radial distribution function? Radial distribution function show the distribution of neighboring atom as a function of internuclear distance from the absorbing atom. For example, this is absorbing atom. So it show the distance, the internuclear distance from the other neighboring atom. For example, here are neighboring atom. So this is radial distribution function. Shells of neighbors known as coordination shells surround the absorbing atom. For example, this is absorbing atom. So other shells which are surrounding this absorbing atom. This is absorbing atom. Other shells of atoms which are surrounding these absorbing atoms are known as coordination shells. And the internuclear distance between the absorbing atom and the surrounding atom, when it is plotted, it is called radial distribution function. And the radial distribution function, when it is obtained, it is fitted to a series of trial structure models, and these trial structure models are available in the literature. So radial distribution of that absorbing atom is fitted on trial structure models available in the literature until a structure which best fit the data is obtained. So that's trial structure model which best fit on this radial distribution, that is obtained. Then it means the structure of that model will be identical to the atoms or substance which we are studying. The final structure which is obtained, we have studied in the previous slide that the radial distribution function is fitted on the trial models. So on which trial model it is fitted, it means the structure of the substance matches to that trial model. So when this final structure is obtained, the final structure will define the number and types of atoms and their distance from the absorbing atom. This is, for example, absorbing atom and other atoms surrounding this atom. So what information will be obtained from the final structure? What type of detail will be obtained from the final structure? Number of atoms, how many atoms are surrounding the absorbing atoms, types of atom, what is the nature of that atom, that is for example either they are iron, zinc, and so on, types of atoms, and their distance from the absorbing atom. Internuclear distance is also refined in that final structure. So three things are defined in the final structure: number of atoms surrounding the absorbing atom, types of atom which are surrounding the absorbing atom, and their distance from the absorbing atom. It is difficult to differentiate atoms of similar atomic number. The atoms which are present here, if they have similar atomic number, means they are isotopes. Isotopes are those atoms which are having similar atomic number but different atomic masses. So if there are two isotopes present, so it is difficult to differentiate the isotopes. And important to note that EXAFS only give data on distance, no angular information is available. Only the internuclear distance is obtained in this data, and no angular information is available. What type of angle is present between the atoms? There is no information about the angle. Depending on the quality of data obtained, distances can be refined to about 1 picometer. Up to 1 picometer, that is up to this small distance, the the small distance can be refined, the small distance can be measured, and in favorable cases several coordination shells out of 600 picometer can be refined. Up to what distance? 600 picometer. There may be a number of shells surrounding these absorbing atoms. So up to 600 picometer coordination shells, they can be refined, their internuclear distance can be measured, and the type of that atom can be measured. This is EXF spectrum for this complex diammonium manganese tetrabromide, and this is a radial distribution for this complex, and when Fourier transform operation is applied on this type of spectrum is obtained. The EXFS data in the figure show the manganese KH of the pair complex. This edge is showing the manganese KH, mean the electron are excited from the K shell of manganese. So due to this excitation, the manganese KH is formed, and we see one coordination pair of 4 bromine atom at a distance of 2.49 angstrom. By interpretation of this graph and by putting the values in different equations, which is a very complex process, we will find that manganese is surrounded by 4 bromine atoms, and the internuclear distance between the manganese and bromine atom is 2.49 angstrom. This is done by complicated calculations corresponding well to the tetrahedral coordination found in X-ray crystal structure. In the X-ray crystal structure, same structure is obtained for this complex tetrahedral structure, and in the EXFS data, tetrahedral structure is obtained. X-rays are very penetrating radiations, highly penetrating radiations. So EXFS, like X-ray crystallography, examine the structure of bulk of solid, because it can penetrate inside the atom and it can reach up to the nucleus. It has the disadvantage that it only provides information on interatomic distance, or internuclear distance is measured in EXFS structure, but has the considerable advantage that it is not confined to crystalline sample and can be used on amorphous solid, glasses and liquids, and which was a drawback of X-ray diffraction analysis, because X-ray diffraction analysis can only be performed for crystalline substances, and the advantage of EXFS technique is that it can be performed for crystalline as well as amorphous substances. Not only that, but by using different absorption edges, it can investigate coordination around more than one type of atom in the sample. We can calculate by EXFS technique, we can calculate, we can find the coordination number of the central absorbing atom, how many atoms are surrounding the central atom. So because different absorption edges are obtained, for example, manganese absorption edge will be on different position, the iron absorption edge will be on different position. So different absorption edges are formed, so we can find the coordination around more than one type of atom in the sample at the same time.